Hi everyone, in this video we are going to discuss about microscopy, principle and applications for the study of microorganisms. Microscopy is the science of investigating small objects using a microscope. Its principle is when a tiny object is placed within the focus of the microscope, a virtual, erect, and magnified image of the object is formed at the least distance of distinct vision from the eye held close to the lens. The applications of microscopes can be seen in various fields like botanical field, biological field, crime investigation areas, medical field and educational field. Microscope resolution is the ability of a lens to separate or distinguish between small objects that are close together. Refractive index is the measure of how greatly a substance slows the velocity of light. A microscope is an optical instrument that uses a lens or a combination of lenses to magnify and resolve the fine details of an object. Microscopes are of two categories, light microscope and electron microscope. In light microscope, magnification is obtained by a system of optical lenses using light waves. It includes bright field, dark field, fluorescence and phase contrast microscopes. An ordinary microscope is called a bright field microscope because it forms a dark image against a brighter background. Example, algae. The dark field microscope is used to observe living unstained cells and organisms. Field appears black and the object is brightly illuminated. Example, Treponema pallida. The phase contrast microscope converts slight differences in refractive index and cell diversity into easily detected variations in light density. It is an excellent way to observe living cells. Example is Pseudomonas. The fluorescence microscope exposes a specimen to UV, violet or blue light and forms an image of the object with the resulting fluorescent light. Next, we can see what is an electron microscope. It is a system of electromagnetic lenses and a short beam of electrons which is used to obtain magnification. They are of two types, transmission electron microscope and scanning electron microscope. Transmission electron microscope TEM is complex and sophisticated. In this, a heated tungsten filament in the electron gun generates a beam of electrons that is then focused on the specimen. Since electrons cannot pass through a glass lens, donut-shaped electromagnets called magnetic lenses are used to focus the beam. The column containing the lenses and specimen must be under high vacuum to obtain a clear image. An enlarged image of the specimen is formed on the fluorescent screen. This is the picture of TEM. Here we can see an electron gun, specimen holder, fluorescent screen, and the final image can be displayed on fluorescent screen or photographed. Next is scanning electron microscope, that is SEM. It produces an image from electrons. It is used to examine the surfaces of microorganisms. Its resolution is 7 nanometer or less. To create an image, the SCEM scans a narrow tapered electron beam back and forth over the specimen. Surface atoms discharge a tiny shower of electrons called secondary electrons. Secondary electrons entering the detector strike a scintillator, causing it to emit light flashes that a photomultiplier converts to an electrical current and amplifies. The signal is sent to cathode ray tube and produces an image. This is the picture of SEM. Next we can see the components of microscope. First is optical components. It can pass light. They include light source, condenser, objectives, eyepiece and body tube. Non-optical components include stage, base, coarse and fine adjustment, and revolving nose. It cannot pass light. The functions of components. Light source will capture light. Condenser focuses the light. Objectives forms real image. Eyepiece magnifies the primary image produced, and the body tube separates the objective and the eyepiece. Stage is the section on which the specimen is placed. Coarse and fine adjustment raise and lower the stage more rapidly. Revolving nose. Objectives are screwed in a revolving nose piece through which they are changeable. This is all about microscopy. Thank you everyone.